Dear students, let us take up the next lecture on photosynthesis, and in this lecture I will talk about photosynthetic light and dark reaction. Let us start with the light reaction. Light reaction is one of the photosynthetic events that take place in the thylakoids of the chloroplast. During light capturing event of photosynthesis, the absorbed photons excite chlorophyll molecules. Light is absorbed mainly in the antenna complexes formed by hundreds of chlorophylls, accessory pigments and proteins that are located at the thylakoid membranes. Photosynthetic antenna pigments gather the photon coming from sun and transfer the energy to a specialized chlorophyll protein complex known as reaction center. The reaction center contains one or more molecules of chlorophyll A with multi-subunit protein complexes. The antenna complexes and the reaction centers are integral components of the thylakoid membrane and form the photosystems. The reaction center of the photosystem initiates a complex series of chemical reactions that capture energy in the form of chemical bonds. Plants and some photosynthetic prokaryotes have two reaction centers, photosystem 1 and photosystem 2, that function in series. The two photosystems are spatially separated. Photosystem 1 is found exclusively in the non-stacked stroma membranes of thylakoids and photosystem 2 largely in the stacked grana membranes. The chlorophylls of photosystem 1 reaction center absorb maximally at 700 nanometer and represented by P700. Those of photosystem 2 reaction center absorb maximally at 680 nanometer and represented by P680. Photosystem 2 and 1 carry out non-cyclic electron transport, oxidize water to molecular oxygen and reduce NADP plus to NADPH. The photosynthetic oxygen evolving system is the only known biochemical system that can oxidize water, as it is energetically very difficult to oxidize water to form molecular oxygen. This process provides almost all the oxygen in Earth's atmosphere. Almost all the chemical processes that make up the light reaction of photosynthesis are carried out by four major protein complexes. That is, photosystem 2, the cytochrome B6F complex, photosystem 1 and the ATP synthase. The electrons ejected from chlorophyll travel through a series of electron carriers organized in the Z scheme. Plants use photosystem PS2 and PS1, first one and then the other, to produce both ATP and NADPH. This process is called non-cyclic photophosphorylation because the path of the electrons is not a circle. The electrons ejected from the photosystem 1 and photosystem 2 end up in NADPH. Both photosystem 1 and 2 have antenna complexes and reaction centers and provide excited electrons to primary electron acceptors. However, each photosystem has slightly different enzymes and other proteins associated with it, so each does a slightly different job. In actuality, photosystem 2 occurs first and feeds its excited electrons to photosystem 1. One special feature of photosystem 2 is that there is an enzyme in the thylakoid membrane responsible for splitting water molecule into protons and oxygen. The oxygen formed during splitting of water is released in atmosphere and the electrons of the hydrogens are used to replace the electrons that had been lost by the chlorophyll. The remaining hydrogen ions or protons are released to participate in other reactions. Manganese is an essential cofactor in the water oxidizing process. The connection between photosystem 2 and photosystem 1 involves the transfer of electrons from photosystem 2 to photosystem 1. These electrons are important because photons from sunlight are exciting electrons in the reaction center of photosystem 1, and the electrons from photosystem 2 replace those lost from photosystem 1. In photosystem 1, light is trapped by pigments and the energy is absorbed in the same manner as in photosystem 2. However, this system does not have the enzymes involved in splitting water. Therefore, no oxygen is released from photosystem 1. The high-energy electrons leaving the reaction center of photosystem 1 make their way through a different series of oxidation-reduction reactions. During these reactions, the electrons are picked up by NADP+, which is reduced to NADPH. Thus, the primary result of photosystem 1 is the production of NADPH. Overall, the captured light energy is used to reduce NADP+ to NADPH. Another fraction of the captured light energy is used for light-dependent ATP synthesis, which is known as photophosphorylation. The NADP and ATP formed during light reaction will provide energy for carbon reduction during light-independent reaction. The primary electron acceptor for the light-energized electrons leaving photosystem 2 is a quinone molecule, and when reduced to plastoquinone is a strong electron donor. This plastoquinone passes the excited electron to a proton pump called the B6F complex embedded within the thylakoid membrane. The location of the two photosystems at different sites on the thylakoid membranes requires that at least one component be capable of moving along or within the membrane in order to deliver electrons produced by photosystem 2 to photosystem 1. The cytochrome B6F complex is distributed equally between the grana and the stroma regions of the membranes, but its large size makes it unlikely that it is a mobile carrier. Phaeophytin and 2 plastoquinones are electron carriers between photosystem 2 and the last cytochrome B6F complex, which is a large multi-subunit protein with several prostatic groups. Plastocyanin is the electron carrier between cytochrome B6F and photosystem 1. The electron carriers that accept electrons from photosystem 1 are very strong reducing agents, and they include a quinone and 3 membrane-bound iron-sulfur proteins known as bound ferridoxins. The electron flow ends with the reduction of NADP plus to NADPH by a membrane-bound ferridoxin NADP reductase. Some of the cytochrome B6F complexes are found in the stroma region of the membrane, where photosystem 1 is located. Under certain conditions, cyclic electron flow from the reducing site of photosystem 1 through the B6F complex and back to photosystem 1 is known to occur. In this process, electrons ejected by photosystem 1 can be passed from one of the iron-sulfur proteins to a B-type cytochrome and from there to a plastoquinone pool between the two photosystems. From the plastoquinone pool, electron returns to P700 via the V6F complex and plastocyanin, which is a water-soluble electron carrier. The Q cycle that is included in this cyclic electron pathway results in proton translocation in thylakoid membranes and thus contribute to the electrochemical gradient that drives ATP formation. This cyclic electron flow is coupled to proton pumping into the lumen, which can be utilized for ATP synthesis but does not oxidize water or reduce NADP+. The formation of ATP through this process is known as cyclic photophosphorylation. Cyclic electron flow is especially important as an ATP source in the bundle sheath chloroplasts of some plants that carry out C4 carbon fixation. Next, let us take up the photosynthetic reaction, that is the carbon fixation or dark reaction. The conversion of solar energy to the chemical energy of organic compounds is a complex process that includes electron transport and photosynthetic carbon fixation. The carbon fixation event, also known as the Kelvin cycle, takes place within the stroma of the chloroplast. These stroma reactions were long thought to be independent of light and, as a consequence, were referred to as the dark reactions. However, because these stroma-localized reactions depend on the products of the photochemical processes and are also directly regulated by light, they are more properly referred to as the carbon fixation reactions of photosynthesis. The NADPH and ATP formed during photochemical oxidation of water to molecular oxygen during light-dependent reaction provides energy, electrons and hydrogen needed to build large organic molecules during carbon fixation reaction. So, the materials needed for the carbon fixation reactions are ATP, NADPH, carbon dioxide and a 5-carbon starter molecule called ribulose bisphosphate or RUBP in short. The first two ingredients, that is ATP and NADPH, are made available from the light-dependent reactions. Carbon dioxide molecules come from the atmosphere and RUBP starter molecule is already present in the stroma of the chloroplast from previous reactions. The fixation of carbon begins with carbon dioxide combining with the 5-carbon molecule ribulose to form an unstable 6-carbon molecule in presence of enzyme ribulose 1,5-bisphosphate carboxylase oxygenase or Rubisco located in stroma. This reaction, in which carbon dioxide is incorporated into carbohydrates, was first discovered by Melvin Kelvin and his co-workers in 1950s. All photosynthetic eukaryotes, from the most primitive alga to the most advanced angiosperm, reduce carbon dioxide to carbohydrate via the same basic mechanism through the photosynthetic carbon reduction cycle. In this Kelvin cycle, carbon dioxide and water from the environment are enzymatically combined with a 5-carbon acceptor molecule to generate 2 molecules of a 3-carbon intermediate. This 3-carbon intermediate or 3-phosphoglycerate is reduced to carbohydrate by use of the ATP and NADPH generated photochemically during light-dependent reaction. The cycle is completed by regeneration of the 5-carbon acceptor or ribulose 1,5-bisphosphate abbreviated as RUBP. So, the most oxidized form of carbon, that is carbon dioxide, is reduced to 3-phosphoglycerate, which is further reduced to glyceraldehyde 3-phosphate product. The Kelvin cycle proceeds in 3 stages. The first stage is the carboxylation of the carbon dioxide acceptor ribulose 1,5-bisphosphate, forming 2 molecules of 3-phosphoglycerate, the first stable intermediate of the Kelvin cycle. The second stage is the reduction of 3-phosphoglycerate, forming glyceraldehyde 3-phosphate, a carbohydrate. The third stage is the regeneration of the carbon dioxide acceptor ribulose 1,5-bisphosphate from glyceraldehyde 3-phosphate. In the first phase, carbon dioxide enters the Kelvin cycle by reacting with ribulose 1,5-bisphosphate or RUBP to yield 2 molecules of 3-phosphoglycerate, which is catalyzed by the ribulose bisphosphate carboxylase or oxygenase, which is Rubisco. This Rubisco enzyme has both carboxylase as well as oxygenase activity, in which oxygen competes with carbon dioxide for the common substrate ribulose 1,5-bisphosphate. Rubisco is reportedly the most abundant enzyme on earth and it represents up to 40% of the total soluble protein in most leaves. Large amount of Rubisco is required as it works very slowly by fixing only 3 molecules of its substrate every second. Carbon dioxide is added to carbon 2 of ribulose 1,5-bisphosphate, yielding an unstable enzyme-bound intermediate, which is hydrolyzed to yield 2 molecules of the stable product 3-phosphoglycerate. In the second phase, the 3-phosphoglycerate formed in the carboxylation stage undergoes two modifications. In the first step of modification, 3-phosphoglycerate is phosphorylated to 1,3-bis-phosphoglycerate using ATP generated in the light reactions in presence of the enzyme 3-phosphoglycerate kinase. Finally, 1,3-bis-phosphoglycerate is reduced to glyceraldehyde 3-phosphate using the reduced NADPH generated by the light reactions in presence of the enzyme NADP glyceraldehyde 3-phosphate dehydrogenase. In the final phase, the carbon dioxide acceptor, that is ribulose 1,5-bisphosphate, needs to be constantly regenerated. Through reshuffling of carbons from 5 molecules of glyceraldehyde 3-phosphate, 3 molecules of ribulose 1,5-bisphosphate are generated. Thus, Calvin cycle reactions regenerate the biochemical intermediates that are necessary to maintain the operation of the cycle. Some of the glyceraldehyde 3-phosphate is used to synthesize glucose and other organic molecules, and some are used to regenerate the 5-carbon ribulose molecule, so this pathway is a cyclic pathway. When 3 molecules of carbon dioxide are fixed, there is net gain of 1 molecule of glyceraldehyde 3-phosphate. The chloroplast stroma contains all the enzymes necessary to convert the triose phosphates formed during carbon fixation reaction, that is glyceraldehyde 3-phosphate and dihydroxyacetone phosphate to starch, which is temporarily stored in the chloroplast as insoluble granules. The synthesis of carbohydrates like starch and sucrose provides a sink, ensuring an adequate flow of carbon atoms through the Calvin cycle under conditions of continuous carbon dioxide uptake. An input of energy provided by ATP and NADPH is required in order to keep the cycle functioning in the fixation of carbon dioxide. So, in order to synthesize the equivalent of 1 molecule of hexose, 6 molecules of carbon dioxide are fixed at the expense of 18 ATP and 12 NADPH. The Calvin cycle, which has a thermodynamic efficiency close to 90%, consumes 2 molecules of NADPH and 3 molecules of ATP for every carbon dioxide fixed. Coming to the conclusion, light reaction and carbon fixation reactions are the photosynthetic events found in photosynthetic organisms. During light reaction, solar energy is trapped by the photosynthetic pigments found in thylakoid membranes. Photosynthetic organisms have photosystems which are made up of 2 closely linked components, a reaction center and an antenna complex. There are usually 2 photosystems in photosynthetic organisms. That is, photosystem 1 which absorbs light maximally at 700 nanometer and photosystem 2 which absorbs maximally at 680 nanometer. The use of the 2 photosystem helps in harnessing maximum solar energy during photosynthesis. So, during light reaction, solar energy is used to make ATP and the reduced compound NADPH with the release of molecular oxygen. In carbon fixation reaction, the ATP and NADPH formed during light reaction are used for the synthesis of organic molecules. Through carbon fixation, which occurs in stroma, the solar energy is finally converted into chemical energy through many stages to form the reduced organic compounds. These organic compounds are the source of energy for most of the living organisms on Earth. So, understanding photosynthetic events are very important as it is the only process to harvest solar energy by living organisms on Earth. Thank you.